cytosine and guanine pairing can be found in both DNA and RNA hybrid form during replication and transcription. The two nitrogenous bases are held together by three hydrogen bonds. The first hydrogen bond is found between oxygen atom of the keto group at C2 of cytosine and the hydrogen atom of the amino group at C2 of guanine. The second hydrogen bond is formed between N3 of cytosine and hydrogen atom attached to N1 of guanine. The interaction between hydrogen atom of the amino group at C4 of cytosine and oxygen atom of keto group at C6 of guanine is the third hydrogen bond. DNA with higher guanine and cytosine content is more stable than DNA with adenine and thymine-rich regions. Having one more hydrogen bond between guanine and cytosine than adenine and thymine needs more energy to break the nitrogenous bases apart. Thus, the melting temperature is relatively higher when DNA has higher cytosine and guanine content. Cytosine and guanine content is used to predict the annealing temperature of primer to DNA in polymerase chain reaction. Base pairing between adenine and thymine can be found in DNA only. There are two hydrogen bonds holding the two nitrogenous bases together. One of the hydrogen bonds is formed between one of the hydrogen atoms of the amino group at C6 of adenine and the oxygen atom of the keto group at C4 of thymine. Another bond is found between nitrogen atom at position 1 of adenine and hydrogen atom linked in N3. The hydrogen bonds between adenine and thymine are important for DNA to maintain a double helix structure. Since they are not very strong bonds, they can be broken at elevated temperature in DNA replication and transcription. The initiation of these reactions often starts at adenine and thymine-rich sites because the breakage of two hydrogen bonds between adenine and thymine requires less energy than guanine and cytosine. Rich sites between have three hydrogen bonds between guanine and cytosine. The phosphate group is important in living things in different ways. Firstly, it is an important structural component of nucleotide, which is the basic structural unit of DNA and RNA. Secondly, it is a component of energy-rich molecules such as adenotriphosphate. Thirdly, it is also bound to coenzymes like nicotinamide adenine nucleotide phosphate involved in anabolic reactions. It is also a part of hydrophilic head. Deoxyribose is a pentose sugar important in the formation of DNA or deoxyribonucleic acid. Deoxyribose is a key building block of DNA. Its chemical structures allow for the replication of cells and DNA double helix configuration. DNA is pivotal to our growth, reproduction, and health. It contains the instructions necessary for your cells to produce proteins that may affect different processes and functions in your body. Because DNA is so important, damage or mutations can sometimes contribute to the development of disease. DNA is essential to biotechnology and medicine by virtue of the fact that it not only provides the basic blueprints of all life, it is fundamental determinant of how the body functions in the disease process. DNA has helped revolutionize the investigation of disease pathways. It is also critical to the identification of pathogens. It assesses an individual's genetic susceptibility to specific diseases diagnosed.